Cuando nosotros vemos a Latinoamérica, el Caribe y España, no vemos las
When we
fronteras,
see
vemos
Latin
las
America,
necesidades,
Uruguay,
las
and the
oportunidades y
Caribbean,
también las
we
esperanzas
see
compartidas por más de
opportunities,
17
hope,
millones de agricultores familiares.
shares by
Ellos producen
more
la
than
mayor parte de los alimentos
17,000
de la región que
agricultors.
contribuyen a la
This
sostenibilidad
is part
ambiental
of
y la
our friends who
biodiversidad, pero
contribute to
también enfrentan los desafíos de
sustainability in our
sostener y
region.
mejorar sus
But
sistemas
we face
productivos en un
different
contexto de crisis
challenges,
climática a través de la
crisis,
tecnología y la
climate
innovación
change.
aplicadas a los
So
sistemas
through
agroalimentarios.
innovation and
Desafíos sin
technology,
fronteras que requieren
we can
soluciones sin
face
fronteras.
these
Somos
challenges
FONTAGRO,
together.
una plataforma de cooperación
We are
internacional para el
FONTAGRO,
desarrollo,
a platform, a
cofinanciamiento de
regional
proyectos de
fund for
ciencia e innovación
agricultural
agropecuaria,
technology.
fundado hace
We
más
do
de 26
science,
años, para
and we
impulsar
have
la
been
investigación
founded
e
more than
innovación en
20
la
years
agricultura
for
regional,
our innovation
reconociendo su
in the
importancia
region.
prioritaria. En América Latina y el Caribe,
In
contamos con
Latin
una sólida
America and the
estructura de gobernanza
Caribbean,
conformado
we
por un
have a
comité
structure, a
ejecutivo,
body
una
of
secretaría
governors,
técnica,
a board of
administrativa,
directors, a
para unir
secretary
esfuerzos y atraer inversiones públicas y
to
privadas en el
join
sector agropecuario,
investment
formada por una
locally
alianza de
and
15 países miembros
internationally.
con el respaldo
We
del
are formed by
IICA,
15 member
Ministry of
countries,
Primary Industries,
be it
Global
IICA
Research
and other partners,
Methane
Global Research Alliance,
CGA y Alianza
Food
Biodiversidad.
and Agricultural Organization of the United Nations.
Entre los
We
investigadores
do
de los institutos
collaborative
nacionales de
projects
tecnología de
with
15 países de
research
América Latina,
and
el Caribe y
investigators
España.
in different countries in
Contribuido
Latin
a la
America, the
realización de
Caribbean,
204
and
proyectos
Spain.
de innovación, con
We
una
have
inversión de más
conducted
de
204
$150 millones de
innovation
dólares. En la
projects,
actualidad estamos
innovation in
ejecutando 59
$150
proyectos que
million.
conectan los esfuerzos de
We
1.500
conducted
investigadores en
59
campos tan
projects
diversos como
that were
cultivos,
participated
ganadería,
1,500
alimentos enlatados, recursos
researchers
naturales,
in the fields of
producción forestal,
natural
agricultura.
resources,
Somos FONTAGRO,
agricultural.
ciencia
We are
colaborativa en acción
FONTAGRO,
para un futuro sostenible
a
e
platform for
inteligente.
collaboration for a sustainable and intelligent future.
Muy buenos días. Desde FONTAGRO es un gusto
Good
darle la
morning.
bienvenida a este
From
taller
FONTAGRO,
sobre
we
control
welcome you
vegetal
to
del
this
HLB,
webinar
que es una enfermedad que representa
to
uno de los mayores
control
desafíos para la
HLB,
agricultura en
a
general y,
disease
por
that is
supuesto,
the main
para la
challenge in
citricultura
agriculture
en las
for
Américas.
the control of
Durante estos 2 días
citrus
vamos a
greening.
tener
In these 2 days,
unos panelistas de lujo
we will
que
receive
nos van a compartir
some
sus
panelists
experiencias
who
y
will
avances
share
en
their
sus
expertise
investigaciones
and
sobre
advancements in
detección temprana,
research
sobre manejo
on
del
how to
vector,
early
sobre
detect
mejoramiento genético,
HLB,
estrategias de coordinación,
science,
todas con el
coordination
objetivo de
strategies.
fortalecer la capacidad
All
de
panelists
respuesta
will
que
share
tenemos
their
frente
knowledge
a
to
esta
bring an
amenaza.
answer to this
Desde
threat.
FONTAGRO, nuestra misión es promover la
From
innovación
FONTAGRO,
científica y
our
tecnológica
mission
en la
is to
agricultura de América
promote
Latina y el
innovation
Caribe.
in
Apoyamos
agriculture in
proyectos
the
que mejoran
Caribbean.
la productividad y la sostenibilidad de nuestros
We
sistemas
support
alimentarios.
projects,
Este taller
different projects,
refleja esa
and this
visión,
webinar
la
reflects our
importancia de la ciencia
vision, the importance
colaborativa entre
of
distintos países de
collaborative
las Américas
science
y la
among
acción
different
coordinada
countries
para
in the
proteger
Americas
los
and
cultivos, en este
coordinated
caso,
actions to
como en el caso de
protect
esta enfermedad.
our
Para aquellos
crops
que,
in these
for
areas.
those who want to listen to this webinar in English, we'll be recording it and then you can see it maybe tomorrow or in a few days in our YouTube channel. Muchas gracias por acompañarnos y muchas
Thank you
gracias a
so
los
much for
panelistas.
being here.
Y
Thank
le
you to
paso la
the speakers
palabra
for being
a
here today.
Charlie Messina.
And I give the floor to Charlie Messina.
Oops, sorry. Thank you, Martin. There are very few other words of thank everybody to join. This is an important workshop that stemmed from a collaboration and a meeting that we have in FONTAGRO, strategic for both Latin America and the United States. This is an opportunity to collaborate, to come together and create that collective mind that is what is going to be required to find a solution to HLB. From the Crop Transformation Center, briefly, for those who are not familiar with, was created with the aim of hastening genetic gain for many different crops using various technologies. One of the things we learn is that germplasm management and biotechnology has to come together in a way to fight diseases, improve nutrition, and increase productivity. And this is what we are all about. It's about people. It's about delivering solutions for people. For us, it's strategic to collaborate with Latin America. HLB is a problem at continental scale, and you will hear more about that in talks later this week. So with that, I would like to thank all of you for participating, and hope we have a good dialogue and come together at the end with a collective mind so we can advance this science, technology, and deployment to help growers' bottom line and income and well-being. Thank you very much.
Muchas
George?
gracias, Charlie. Bueno, empezamos con nuestro
Thank you,
primer
Charlie.
panel,
We
que
will
es sobre
begin with
detección
our
temprana,
first
diagnóstico y
panel that
vigilancia.
has to do
Vamos a
with
tener
early
tres
detection,
presentaciones y la
diagnostics, and
primera
surveillance.
es,
We will have
está a
three
cargo de la doctora
presentations.
Romina
The first
Gasis.
one is in charge
Adelante,
of
Romina.
Romina Gasis. Romina.
Ok, muchas gracias.
Thank you so much.
Bueno, voy a empezar a decir buenos días con todos en español,
First
pero
of
después
all,
voy a dar la charla en inglés.
good morning
Good
in
morning,
Spanish, and
everyone.
then I will follow
My
up
name
in
is Romina
English.
Gasis. I'm happy to be here. I'm originally from Peru, so I am extra happy to be among colleagues, trying to build these connections with researchers in Latin America. So today I'm going to be presenting about diagnostics. I am with the University of Florida. I am based at the Tropical Research and Education Center. And a little bit about myself, since this is a new group for me, I would like to introduce a little bit my program. I'm an associate professor in the Department of Plant Pathology. And as I mentioned, I'm based at the Tropical Research and Education Center. I'm also the director of the Plant Diagnostic Clinic, which is housed at these centers. I specialize on diseases of tropical and subtropical crops, especially in ornamentals and tropical fruits. And I have a 60% extension appointment and 40% research, and I'm considered an extension state specialist. I work on different crops, and I have worked in the past with cacao, rubber tree, looking at fungal taxonomy and diversity, and also looking for biological control agents to manage diseases. So this is my group. You can find more information in any of those QRs. We work on topics related to plant health in horticultural crops and natural ecosystems, with a focus on tropical and subtropical systems. So I work with different diseases, and that will depend on the funding. And so a lot of my program focuses on avocado, dragon fruit, passion fruit, and I do, depending on the funding and the needs of the growers, I also work with lime, mango, and do exploratory work on other tropical fruits. So I have to say that I do not work with citrus production. Most of the citrus production is located in Central Florida. And before, in the past, in South Florida, where I'm located in Homestead, you can see here at the south of South Florida. And we used to have a lime industry in the past. That is not anymore here. We used to have, back in the 80s, more than 7,000 acres. And I was not here when that was the case, but it was a big industry for us. And it was really important industry because we were known for our key limes. And so, unfortunately, there were several things that made that industry collapse, starting with a Hurricane Andrew, and then there was a canker eradication program, which eliminated a lot of these plantings, and of course, then we got citrus greening. So far, and currently, there's only few plots still present in the area, and there is very low to none commercial production. So, however, when I got to UF, we did have a experiment, and we did have a project on Lyme and trying to understand if we can still produce Lyme in this area under disease pressure, right? So kind of like learning how to live with a disease, and we were trying to figure it out if with the disease pressure that we have, which is pretty high, we can still it's still profitable for the grower using multiple strategies to manage the pest and the disease. And so this is just to show you, that's an experiment plot that we had here at the station. And the experiment starting in 2017 when we planted the trees. And you can see that my lab tested the trees every 3 months for multiple years. And you can see how the first year we almost didn't get any positive, and this is natural infection. And so we didn't get any positive tree, but as soon as we got positive trees, you can see how the disease has spread very fast throughout the plot. And in few years, so starting 2018 all the way to 2020, in 2 years, the entire plot, 239 trees, tested positive. So this is just to show you that this disease spreads really fast, and we know that. But this is kind of like a visual representation on how this disease can really take all the trees in a plot. And so this is just to show you how severe this disease is. So today I'm going to be talking about diagnostics, and I'm sure you are aware of this, but diagnostics is central to an IPM program. So before we take any action, before we start implementing any management practice or even designing a plan to address the problem, we really need to understand what is causing the problem in the plant. It can be abiotic, it can be abiotic. And so we really need to determine what's the causal agent of the plant decline or plant damage or plant injury. And so diagnostic is really central to plant health. And so this is the reason I'm going to be talking about diagnostics. And so citrus greening or HLB, as you know, can be caused by several agents. And so there's 3 species of Liberibacter. We use the word candidatus before Liberibacter because that just is saying that this bacteria is unculturable. And so this is important. Understanding that there's 3 causal agents is important for diagnostics, right? Because I'm going to explain you later, but there are different tools that we can use to identify these bacteria, especially the DNA-based tools. They will be able to distinguish, there will be specific tools to distinguish each of these species of bacteria. There's also, and then there's different distributions of these bacteria. So you can see here that for our purposes, we have in Latin America the Liberibacter asiaticus, right? Although in Brazil there seems to be more than one species of bacteria, but the most commonly found is the Liberibacter asiaticus. That doesn't mean that we should not be prepared to test for the other bacteria, right? Because as there was an introduction of these bacteria from Asia, there could be a second or a third introduction of a different species, which could complicate diagnostics, right? So we should be prepared to identify the 3 causal agents of the disease. And there's different vectors as well, which are distributed differently throughout the world. But in case of Latin America, again, we have the Diaphorina citri as the main vector of the disease. And again, we should be familiar with the other vectors, and we should be understanding which are the diagnostic characters of those vectors. And so how do we diagnose diseases, right? So depending on the uniqueness of the symptoms they induce in their hosts and the structures they produce, diseases can be diagnosed in several ways. Some can be diagnosed in the field if the person doing the diagnosis has had a lot of experience with the host. But remember that also the environment plays a role in how these symptoms are manifested. And so the person has to be experienced, have to have seen the disease in multiple regions and in multiple hosts. And maybe that person will be able to do an accurate diagnosis in the field. And while other diseases may require more specialized tools and expertise. So this is kind of like a breadth of tools that we use to diagnose diseases. Of course, everything starts with seeing the symptoms and or the signs in the field. But also we as plant diagnosticians or diagnosticians and the ones that are running diagnostic clinics, we sometimes cannot see the symptoms in the field and will depend on that sample submission to do our diagnostic. So not always we have the option of observing those symptoms in the field. Because we are dealing with an unculturable bacteria, right? That's why we use the term candidatus. So that means that these bacteria cannot be cultured. And because bacteria in general, they lack easily discernible diagnostic features. So there's no many morphology, not much morphology that we can base our diagnosis on, especially at the species level. Diagnosis can be challenging. Can be challenging for bacteria in general and can be extra challenging for bacteria that you cannot culture. And so some of the traditional methods that we use in plant disease clinics, like microscopy or culturing, or even just incubating that tissue to see the signs of the causal agent. We can do a lot of that with fungi, for instance. We will put those pieces of infected wood or leaves or any tissue that is infected, and we incubate that very easily in a homemade moist chamber, and then you can see the fruiting bodies or the structures coming out of that infected material. So we cannot do that with citrus greening, obviously, and so that will limit the number of tools that we have available. And so these are the tools that we have available for diagnosing and monitoring citrus greening. So if we have the chance and the opportunity to see the symptoms in the field, of course, that's preferable. And the symptoms may be reliable only when trees are located in regions where the disease is endemic, such as Florida. I think that a lot of my colleagues can go to the field and say, well, this tree certainly has citrus greening because we are used to see them. We are in a region that likely all the trees have it. And so it's kind of easier to say it's easier to do a diagnosis in the field in regions like this. However, for regions that the disease is not present yet, or that in an orchard that perhaps hasn't been infected yet. So those are the cases that you really need to do some lab testing to be sure that the disease is present and the causal agent is detected. So observing disease patterns also within individual trees and across a planting is really helpful, as we are going to be talking later. There are some symptoms of nutrient deficiency that look very similar to the symptoms induced by the pathogen. And so plant pathologists use a lot of field observations, especially looking at the distribution of the symptoms within a tree, within the canopy, and in a plot. So diseases usually do not affect all the trees at the same time. at the same disease progression and severity, right? So we are looking for patterns. If we see that all the trees have the same symptoms and even all the leaves or most of the leaves in a canopy have the same symptoms, then we are thinking more like abiotic, like maybe a nutrient deficiency and so on. And so really understanding and learning the symptoms in the field, including not only the symptoms on one leaf or one tree, but looking at the whole picture, understanding and observing what's the distribution of the symptoms in the field is very important to make a decision. And so that being said, here you can see HLB infected foliage, which typically shows that blotchy mottling on the leaves. You can see that some part of the leaf have like really dark green blotches, and then you have background of yellowing, and you can easily see that the pattern is asymmetrical, right? I would call these really textbook symptoms. Not always you will see this, right? And that's something that I always try to convey to people, that when you look online or when you consult a book, they usually show you the textbook perfect photos, right? And so the symptoms sometimes don't look like that in the field because you have multiple things going on. The environment is playing a role and the symptom expression. There may be other things going on like abiotic disorders or other diseases. You know, you may have canker on top of citrus greening infected foliage, so it won't look that perfect, right? But I wanted to start showing you kind of like the textbook symptoms that you would see on the foliage. And this is Lyme. Here are several other symptoms. So this disease can cause symptoms on the foliage, on the fruit, and eventually is lethal for the tree. But it's a slow decline, and that depends on the citrus species that you are dealing with. Lyme is a little bit more tolerant or less susceptible. It doesn't die that fast as other type of citrus. But you will find kind of the same symptoms, right? On the leaves, you will find that blotching, and sometimes you have deformed leaves, like you can see at the tip that kind of twist. And the photo at the center is a abiotic, just to compare how do you see the symmetrical pattern. So if you have the midrib here, and you can see that on both sides, it looks almost like a mirror, like identical, right? But at the right, you can see the blotchiness that an HLB infected leaf shows. This disease also causes damage to the fruits, and so it can cause deformities in the fruit, abnormal shapes, premature drop, reduced size. Also, it reduces the quantity and quality of the juice of these fruits, and can cause this greening, right, on the fruit, which, you know, the fruit doesn't really change color like it should. And when you do a transversal cut, you can see that the midsection, the column is deformed. And so that's good indication that you have citrus greening. Now, because the photosynthesis capacity is reduced, and I'm sure you are going to be hearing more about the effect of these bacteria to plant health in general, you are going to get symptoms like dieback, like defoliation, dieback. The tree loses vigor and it becomes like a chronic disease. And in a matter of 2 years, 3 years, depending on which citrus you are dealing with and depending on the management of the tree. So if the grower is pumping a lot of fertilizers, the tree may hang in for a while longer. But these are some of the symptoms that we observe on the Lyme plot, and you can see that You see the dieback, the defoliation, and eventually the death of the tree. And so in our case, we found that the trees died within the 3 years that we started the experiment. So the symptoms on the fruit, at least when we talk about Lyme, can vary quite a bit. And so you have here several examples of the symptoms that we observe on the fruit. And even when you see the symptoms on the leaf, that tree is clearly infected with HLB and it was confirmed through laboratory testing. Some of the fruit did not show any symptoms. Now, I'm not going to really give you the details of this experiment, but at the end, when we harvested all the fruit, we evaluate the quality of the juice, and even fruit that may look okay, the quality and quantity of the juice was not really comparable to a non-infected tree. And so even if the symptoms, external symptoms are not present, there may be an effect on the quality of the juice. But in any case, you can see here, there's some yellowing and stripes on the fruit. And then you can see again this blotching and some of the dieback on the tree here. And so as I mentioned, other diseases and disorders can cause similar symptoms. And here you can see the comparison between a symmetrical pattern, which is caused by a nutrient deficiency, and the asymmetrical pattern, which is caused by HLB. Another symptom that you will see is the starch accumulation. People don't do this anymore as a diagnostic, but in the past, it was one of the tools that people can use in the field, which is using iodine to dye the starch that accumulates when you have an HLB infected tree. So I wouldn't say that is a definitive diagnostic tool, but it definitely can suggest that you have that disease. And as you know, not all psyllids carry the pathogen. So even if you see the psyllid, that doesn't mean that the disease is present in that area. And we have experienced that in California, where the psyllid was found without the bacteria being present. And so moving along with other now more lab-based tools. So we are going to talk about the serological and DNA-based techniques, which are performed on a sample. So that can be people submitting samples to a plant clinic, to a lab, to a colleague, to a university. So let's see that we are not in the field to take notes on disease, symptom, distribution, incidence, severity, patterns. We are not able to do that. We are just getting a sample, right, through the lab. And so sampling for HLB diagnosis can be challenging due to the low titers during early infections, and that happens to a lot of diseases, and the uneven distribution of the pathogen. So sometimes when we hear about vascular pathogens, we hear that these diseases are systemic. They don't mean that they are 100% systemic, right? Especially when we are not talking about viruses. And so even laurel wilt, HLB, those are two vascular diseases. One is distributed in the xylem, like laurel wilt, and HLB is in the phloem, right? So even though they migrate and they move from the infection site all the way to the roots, their distribution is uneven. And so that makes sampling challenging. And that's a very common challenge that we have as diagnosticians, is to teach people collect good samples, right? Because our diagnosis is definitely dependent on how good the sample is. And so lucky for us, there was this study that quantified the amount of Liberobacter asiaticus in a plant, right? And so you can see here, we have a potted plant on the left, And then we have also the distribution of the pathogen in the fruit. And the authors use qPCR to quantify the amount of bacteria present in the different tissues. And so you can see that the red and pink colors have more amount of bacteria, more inoculum than the colors that are more blue and green. So first thing, you can see that it is very uneven, right? Definitely the bacteria moves and multiplies and colonizes different tissue systemically, but the distribution is uneven. And in the fruit, you can also test, which is a good thing, but also the distribution varies according to the part of the fruit and the tissue in the fruit, and also in the species, right? Or the variety of citrus. So you have different accumulation depending if you are dealing with lime or you're dealing with lemon. And I would think that it's the same with the oranges. So you have different amounts of bacteria. However, we usually recommend to sample the midribs. So that's what my lab does. We ask to take samples not from the new growth, because that's where the psyllids are. If you are hunting for psyllids, then psyllids love the new growth. And so you can look for psyllids over there, but then the higher amount of inoculum, which will make your diagnostic tool more successful, then it would be the kind of mid flash. And so the older flash, also the older leaves can also have a good amount, and we use the midribs. Those are the tissue, that's the tissue that we use for the testing. And again, symptomatic samples are always the best samples. And so there is no perfect diagnostic tool, number one. And to have a diagnostic tool that can detect the pathogen before symptom onset is the holy grail of any diagnostician. There are not many of those. Most of diagnoses, even in the lab, are based on symptomatic tissue, and which, you know, it's a challenge, right? We can test early. Perhaps there are some tools that can detect early infections, but then you always have the risk of false negatives, right? So something to consider. If you can submit samples that have already the symptoms, those are the best. Always include a positive control. So try to get DNA from known, confirmed, infected host material. And of course, negative controls are also should be included in the test. And something to consider is that when you use excessive host tissue, that can interfere with DNA extraction and reduce the efficiency of PCR, qPCR, or LAMP, and serological tests. So any of the tests that we do in the lab, we always have to be mindful of the amount of tissue that we use, because that we are doing direct detection, right? We are not culturing, because remember, these bacteria cannot be cultured. And so because we are doing direct detection, we are dealing with the background DNA, proteins, carbohydrates, and all of those molecules can be inhibitors. Not only can clog the columns that we use in DNA extraction, but can also inhibit some of these detection reactions, right? Either the serological detection or the DNA-based detection. And so again, I always ask for symptomatic tissue. And you can see clearly here in this photo how the new growth, yes, it does look yellowed, but it has none of the blotching. However, the previous season flash, you can see here the blotching and the thickening, how the midribs are really thick, and the tips a little distorted. So those leaves are the best sample for us to do the testing and take to the lab. Now, there's so much resources out there for diagnostics of HLB. So as I mentioned before, in my area, we used to have Lyme, And we used to do lots of diagnostics in the past. In the recent years, since we don't have much commercial production of lime, we are only dealing with homeowners and their citrus trees. So we don't do a lot of diagnostics now. So I may not be the best person to answer all the questions, but there is a lot of information out there, and there are a lot of information on different DNA extraction kits, different tissues and protocols to test for HLB, like this one that is showing that roots are good tissue sample for testing. And so I will recommend you to look into the literature and compare DNA extraction protocols and what's the best tissue for your lab to use for testing. And so moving along with the serological test, we all know ELISA. ELISA is a very commonly used diagnostic test for human pathogens, for animal and plant pathogens. And so it's widespread, commonly used in labs, quite easy, and it doesn't require much resources like equipment, but it does require reactives and some supplies to do the testing. And so for HLB, I did a lot of literature review before this presentation, and I could not really find a easy to use protocol, and there's no commercial kit available. But there are labs working on getting these proteins and antibodies in the pipeline of a commercial product, which will, eventually and hopefully end up in an immune strip that you can use in the field. Like we have immune strips for a lot of different viruses on vegetables, for Phytophthora testing in the field. And so eventually this research hopefully is gonna promote the industry to develop more easy to use in the field diagnostic tools. But so far for ELISA, there's no commercial kit available. Now, DNA-based techniques, I would say those are the most commonly used. There are multiple primer sets that have been developed for the different Liberibacter species, and some are suitable for multiplex assays, right? As I mentioned before, even though in Latin America, the Asiaticus species is the most common, labs should be equipped to detect the other two species, because of course there can be another introduction, or maybe there are already, I believe, some countries that have more than one strain or more than one species of the bacteria. And so it would be good to have these multiplex protocols that you can detect, the three causal agents. And so the sensitivity of these different protocols will vary depending on the platform that you use. Here is a list, not exhaustive, but just to let you know that there is different platforms. So we can use conventional PCR, we can use quantitative PCR, we can use qPCR based on Cybergreen or qPCR based on TaqMan. One is more sensitive than the other one is more expensive than the other one. But definitely sensitivity is something that you want. But it will have to be a trade-off between the expertise that you have in your lab, the equipment that you have in your lab, and the resources that you have. You can start with a conventional PCR and narrow down the samples, the set of samples that you have, and then go to the next step. You can collaborate with universities that may have this equipment for other reasons. But this is just to let you know that there are a lot of primers out there that can identify all of these causal agents through different platforms. And they are based on different regions, different genomic regions. And so there's a lot of literature out there for you to check. So the gold standard, I believe, is the qPCR. That's quantitative PCR, and that can be based on cyber or TACPAN. And so those are the tests that most of the labs that are dedicated to diagnose HLB use. Now, my lab, since we really don't work much with citrus screening anymore, we use conventional PCR. So these are very old primers, but they are pretty solid and robust, I would say. And we use a primer that is that focus on Asiaticus. However, we have the other primers in case we need to, in case we see the symptoms and this reaction is negative, then we can use the other primers to see if we have a different causal agent present in the sample. So this is just
Romina,
yes,
Romina,
yes.
te pido por favor si puedes apurar un poquito porque
Okay.
está extendiéndose más de lo previsto la charla.
Okay, sorry. So this is just
Gracias.
the process of the sample. Again, use the midrib, and then the homogenization step is very, very important because the tissue is very fibrous. And so you really need to macerate it quite a bit to be able to detect the DNA from the bacteria that is inside this vascular system. And then you run the PCR and you will see a band or no band. These are a specific primers, so you don't have to sequence, but you can send that product to sequence in case you want to confirm. And you can also use ciliates, which are great samples. If they are infected, of course, they are great samples because they are soft. They have a soft body. And so those are usually don't give you a lot of problems with the extraction. So commercial kits, you don't have to reinvent the wheel. You can go and buy the kits for these LAMP assays. Those kits are based on LAMP assays, which are isothermal amplification systems. And you can do that in your lab or you can do that in the field, but you will have to buy the equipment. And so these are two options. One is from Agdia and the other one is from Diagenetics. And again, those are easy, ready to use reactions, but then you will have to have the equipment to visualize the fluorescence of a positive amplification. So since I am running out of time, I'm not going to cover all of these. However, there are other methods out there based on volatiles secreted by the host that is infected, right? Is secreting these volatiles that can be captured by equipment. There is remote imaging, and based on the reflectance of an infected versus a healthy tissue, foliage tissue. And of course, those have been kind of implemented in AI algorithms so they can monitor. It's mostly monitoring. So I wouldn't use those for diagnosis, but they use it for monitoring the spread. And when you have an orchard that is already infected, you want to visualize which trees are infected so you can remove them or perhaps implement the strategy that the grower is using. And so just to finish, I do want to share this website from the European Union called Pre-HLB, which I think is fantastic. So it has a lot of information on how to be prepared. They even do kind of exercises on what will happen if they have a detection in one of the orchards in a region that HLB is not present. And so they have all the action plan on what to do, who to call, and what strategies to use. And so I would really recommend that you guys check this website. It's pretty nice. And the University of Florida, of course, has tons of resources on citrus greening and detection, management, what are the current methods that growers are using, what's coming in the pipeline in terms of breeding and injections and so on. So I would really recommend you to look at all these educational material. And with that, thank you for listening. And if there's any questions, I'm happy to answer. And you can contact me if you want some of these resources. Thank you.
Muchas gracias, Romina, por esta excelente presentación.
Thank you so
Tenemos una
much,
mucho mejor
Romina,
idea
for your
sobre las opciones de
presentation. We
diagnóstico
now
a
have
partir
a
de tu
better idea
presentación.
about
Las preguntas las
diagnostics
vamos a
after your
manejar al final
presentation. We
de
will
toda la
deal with
jornada,
questions at the end
allí donde vamos a hacer un
of the
poco de
webinar.
discusión y de
We will
intercambio.
have some time for debate
A
and to
continuación,
exchange
tenemos la
information.
presentación
So
de Rick
now
Dantzler,
we will have the
a quien le
presentation
damos la
of
bienvenida
Rick
para
Dantzler.
darnos su
Welcome,
presentación.
Rick, and we give you the floor.
Well, thank you very much. It's a pleasure to be here. Let me see if I can share the screen.
Perfect.
Okay, good. Well, thank you. I really appreciate the chance to be with you today, and I congratulate FONTAGRO and the other sponsoring organizations for pulling this together. I am all about collaboration, and if by having this we can find ways to work together, that would be fantastic. A good outcome from my perspective. Our panel slot is about early detection, and as Romina just shared with you, positive finds of HLB spread very quickly in groves. From the CRDF perspective, we assume that every tree is infected in Florida. That's how we fund our research. And that is not to say that the other specialties are not important, because they are. I don't advocate not funding specialties that deal with areas that I'm not going to mention today, but because we were in such a dire situation in Florida back in 2021 when we seemed to really fall off a cliff, and frankly, because CRDF's funding was declining, we had to necessarily be more focused on what we were going to fund. But I have to tell you now, after CRDF has funded 550 projects and spent several $100 million, and the United States government and other funding entities from across the world have spent record resources on trying to defeat this disease, I have never been more confident. I am absolutely convinced that we are beginning to emerge from a long, dark tunnel. That is not to say that I do not recognize that we have not lost an awful lot of growers, because we have. We only have about 25% of the growers that we used to have. We only are farming about 25% of the land. If you look at how much production we had back at about the turn of the century, it was around 250 million boxes. We're down to 12. Yes, you heard that correctly. Not all of that, of course, is because of HLB. It seems like the whole world is trying to move to Florida, and we are growing so fast that we are rapidly losing citrus acreage. In fact, I am convinced that the biggest threat to our industry long term is not going to be being able to grow citrus in the face of HLB, but to have land to grow it on. Nevertheless, I am convinced that we are going to make it. Now, our industry is not going to be nearly what it once was, but it is going to be several times more than what it is now. I don't think there's any question about that. And I think I am beginning to believe a shift towards other kinds of fruit that we'll get into perhaps during the question and answer session. But let me see if I can advance this slide. Audrey, I can't advance this slide. I'm sorry for having to call in help, but it's just the way it is.
No problem.
Okay. Okay, close that door if you would. Let me go into a couple of the reasons why I am so confident. And I have to say that this first one, the fullness of time, is something that I did not completely appreciate when I started this job 6 years ago. As you know, it takes a long time to get answers in citrus research. It's not like a vegetable where you can plant a seed and find out 120 days later if your experiment worked. It takes years to get answers in citrus research, but our science has now ripened to the point where clear separation is beginning to appear between winners and losers. We see it the most in the area of citrus breeding, and I'll comment on that later, but it really spans the whole gamut of our experiments. Even when you have an experiment that didn't work, you've learned something from that, and it makes you better over time. Also, the appearance of our trees now is fantastic. There are some pockets that are still obviously struggling, but our trees are looking really good now. And if we can get through this season without a major hurricane, I think we are going to see a tremendous rebound in productivity in each of our trees. And of course, the appearance of these trees has created a new enthusiasm in the industry that we haven't had in a number of years. We've had 3 major hurricanes in the last 4 years. Were it not for those storms, I think we might already be several times more than what we are now in terms of production. But nevertheless, it is what it is, but enthusiasm is extremely high. Well, what is the research strategy that we have been implementing at CRDF? It's different than it used to be. When we started, when we knew so little about the disease, 100% of our projects dealt with basic research. But as we learned more, we began to shift our research portfolio more towards applied science. And then when we fell off the cliff in the year 2021, we realized that everything we were about had to be about keeping growers in business for the next few years, at least the next 3 or 4 or 5 years, maybe the next 7, until we can end up with a tree that is going to put this disease behind us once and for all. So this is our research strategy. As limited as it is, but it's exactly where we thought we needed to be. And frankly, we believe it's working because we are seeing leaf canopies restored, we are seeing root regrowth, and these are all precursors to more and better fruit. But our research strategy is to reduce sea loss as much as possible, as efficiently as possible, for as long as it takes to get the tree of the future into the hand of growers. Again, we had to necessarily fall back into this research strategy, and we now have 62 projects under contract, most all of which revolve around this strategy, and we have 10 other projects under consideration, most of which deal with this strategy as well. So that's what we've been working on, and what has been the mechanism for implementing this strategy. Leading the pack has been the injection of oxytetracycline, which of course is an antibiotic. That was the one that we felt that we could get approved the quickest. And it was approved in record time, and it has worked. Now, I will be the first to tell you that not everyone has had the same experience from the use of this therapy. But as my president, Morgan Porter, once said, if 5 of us joined the gym tomorrow and we worked out the same way for the next year, we wouldn't all look the same because we were all starting from different places. And that's what we've seen in the citrus industry. Not all of our trees were starting from the same place, but I think almost every grower will say that they have seen an increase in tree health from the use of oxytetracycline. I'll be the first to admit that we have an awful lot riding on this third year of oxytetracycline, and it has to work better or we are going to have a real problem on our hands. And when I say it has to work better, it has been working in this first 2 years, but many growers have not seen the kind of increase in fruit quality and yield that they were hoping to see, but I'm convinced we're going to see it during this third year. We know, though, that this was always going to be a 5 to 7 year therapy at best, and that we were going to need something else. We also wondered if the tree of the future was going to be ready in this amount of time. We were worried about resistance that might develop to the pathogen, to the oxytetracycline itself. And perhaps there could have been other reasons why we were no longer able to use OTC. So largely through the Grow First program, we have been trying to find replacement antimicrobials for oxytetracycline if that becomes necessary. You are going to hear from Michelle Heck later in the agenda. She has been leading the team that's working on the Grow First project. They have identified many molecules that work as well or better than oxytetracycline. Some require a pretty heavy lift from a regulatory standpoint, and those are primarily other antibiotics, but some do not require much of a lift at all, and some we believe growers could begin using right now. So we have a rotational partner, we believe. That's still being validated to some extent, but we believe that we will be able to have a replacement if necessary, but we want to stay with this third year and see where it goes from there. The university has also been doing some groundbreaking research on the use of plant growth regulators. Doctors Tripti Vashith, Fernando Alvarez, and others have been doing work with PGRs and IPCs, individual protective covers, and brassinosteroids. We are synchronizing flushing and sticking fruit better than we ever have with the knowledge that we've gained from this science. That's going to help a lot as we try to reduce pre-harvest fruit drop. Of course, I think most people believe that long-term, the tree of the future is going to be what saves the industry long-term. And this is also the one that takes the longest to figure out if it's working or not. But again, in this fullness of time and over the many generations of grafting and crossing and doing everything that needs to be done to have a successful plant breeding program, we are beginning to see the benefits of all these efforts. Let me mention just a few projects in the area of conventional plant breeding, if I could. I am absolutely convinced that our breeders have created more tolerant rootstocks and scions than what most growers are currently using. What we need, though, is to validate them sufficiently to give growers the confidence to go all in on them. So CRDF is doing what it can to help out with this. We also are funding a project with Coca-Cola where Coca-Cola took 550 accessions that have been created by the great breeders that we funded over the years, and they narrowed that down to 68. And we are funding a project with them where they are going to do stage 2 field trials on these 68 primarily scions, and most of them are orange-like scions. They're not citrus sinensis. A few are, but most aren't. But I personally think that orange-like hybrids are going to be part of our future. Nevertheless, Coca-Cola is working with our breeders, and we are going to. I really think we are maybe one cross away from having many of these accessions exactly where we want them to be. CRDF has also funded what it calls a grand plan. We're a little over a year into this. It was a 3-year funding effort for the universities plant breeding program and the USDA's plant breeding program, where those programs are working together on bioinformatics and a number of other things. So this is going to provide important funding for these programs to sustain themselves. There is another project underway a proposal that is being considered that I want to mention that I'm personally committed to, and that is to take let me say it a different way, and that is to take the most promising scions that our breeders have come up with and graft them onto the most promising rootstocks. It ran into a little bit of pushback at the CRDF level, so we're trying to get this funded by HLB MAC. So if any of you have any influence with any of those folks, I'd appreciate you weighing in on it. I think that's going to be an important project to see funded. I personally believe that the most gain that we have to make is going to be through gene editing. Doctors Fred Gamitter and Charlie Messina are on the panel, and you're going to hear what they've been doing in that area. But let me just highlight a couple of projects that are underway that revolve around a number of trees that have been created by our biomolecular scientists. One is by Dr. Zonglin Mao from the University of Florida. He is overexpressing the NPR1 gene. It's a transgenic, but nevertheless, he's had trees in the ground since 2019. I haven't seen them in a year and a half, which frankly, they didn't knock my socks off then. They seem to be struggling with juvenility a little bit, but I'm told that they are loaded with fruit this year. And the fruit is good. If there's been a criticism, it's that they're too sweet. I mean, what a great problem to have for a change. But he and Dr. Eric Triplett from the university are in the process of trying to get this approved by the regulatory authorities. We've had numerous calls in the last month trying to find a way to make this happen. So this is a tree that I'm very enthused about. I've seen 50 of these trees in a greenhouse. They've been exposed to hot silage for a couple of years now, and they appear to be HLB symptom free. Dr. Manjul Dutt, he's been grafting non-transgenic scions onto transgenic rootstocks. I think this is a really exciting proposal. Believe it or not, I'm told that we don't yet know that a transgenic rootstock that stays essentially CLAS free is going to bestow tremendous benefit on non-transgenic scions that get HLB and have plenty of CLAS in them. It makes all the sense in the world to believe that they'll work. I believe they are going to work. But we need to get these trees in the field as quickly as we can. He's been hard at work trying to get these trees ready. Soilcia is a private company that acquired the intellectual property rights to many of the genes that Dr. Nian Wang identified. They have a transgenic rootstock that has non-transgenic scions grafted onto it, and it looks fantastic. I saw it about a month ago, and it's young. It's only 3 years old, so we don't know what kind of fruit it's going to grow and whether the fruit's going to be good and all those sorts of things. But at least how it compares to the control trees, it just looks tremendous. And there's a lot of enthusiasm in the industry for this tree right now because of where it came from and how we believe it's going to work. We've already created numerous non-GM CRISPR edited trees. For example, Doctors Wong and Grosser, they've already developed canker-resistant Hamlin and Valencia non-GM CRISPR edited trees, and they're working on red grapefruit. And Dr. Nian Wong, in just the last 6 weeks, maybe 3 months, I guess, has shown us that he has been able to silence EDS1 and DMR6. And we really, you need to keep your eyes on these two, because this has tremendous potential. Everybody's pretty enthused about this. So keep your eye on that one. You're going to hear more about all of this from Drs. Messina and Gmitter later in the agenda. Okay, there was one other slide that I didn't include because, candidly, I didn't read the email as thoroughly as I should have, but I was asked to include a slide on what short, medium, and long-term actions we felt were needed. So what I would say on short term, we need the 4th year of oxytetracycline usage approved by regulators. It was approved for just 3 years. We're in the 3rd year now. In the event that we don't have anything better that we can get approved, we need to have this 4th year approved. I feel pretty confident that's going to happen, but stranger things have not happened, I suppose. Medium term, we need approval of a new antibiotic. One of the things that Dr. Heck has found is that nothing seems to work better than doxycycline. But I had a little skin cancer cut off my arm a few months ago, and they gave me doxycycline as a preventative to infection. So that's going to be a tough one to get approved, but it works really, really well. The one that works almost as well, and one that should not be that hard of a lift from a regulatory standpoint, is tetracycline. In fact, much of the early research that took place with HLB by growers who were experimenting with different things involved tetracycline. And then in the year 2020, there was a decision made to go to oxytetracycline, I think because they felt like it would be easier to get approved. Nevertheless, that's what I would ask for a medium-term need, another antibiotic. If we could get that, we would absolutely make sure that our industry is going to survive until this tree of the future, whatever it turns out to be, is in the hands of growers. And finally, the long-term need that we have is to validate many of these terrific conventionally bred rootstocks and scions and all these transgenics that I've already discussed, plus others that you're going to hear about later in the program. It's one thing to create them, but it takes a while to validate them. As my respected colleague, Dr. Bill Dawson, says, genes have the trees they need because they need them. And so when you knock one out, you don't really know what's going to happen to the tree. So it takes a while to figure those things out. But the ones we've talked about appear to have no ill effects on trees. So I think that if we can validate these as quickly as possible, growers are going to be very pleased with what they find. I'm happy to answer any questions. I look forward to the question and answer session, and thanks for giving me this chance to speak.
Muchísimas gracias, Rick, por esta visión optimista.
Thank you so
por
much,
cierto,
Rick,
pero
for your
un optimismo
optimistic
basado
view.
en
Thank you,
avances científicos y en
because this is
estrategias
based on
basadas en ciencia que
strategies,
nos hacen
on science,
creer
on
en que
advancements on
esto lo
science.
vamos a
So
poder solucionar
I think we
en los
will
plazos que estamos
have
manejando de estas estrategias de corto,
good
mediano
solutions in
y
the
de largo plazo.
future, in the short term
Para
and medium
finalizar
term
esta
too.
primera parte del panel
To
1,
conclude this
luego vamos a
first
tener
panel,
un breve descanso y seguimos con
we
el
will
panel
then
2.
have a break, and
Vamos a
then we
escuchar
will
a
follow with
Rodrigo
panel 2.
Astete.
But before that, we
Rodrigo,
will
por
listen
favor.
to Rodrigo Astete.
Perfecto, muchas gracias Martín. ¿Me cuentan si se está viendo la
Thank you
presentación?
so much,
Perfectamente.
Martín. Can you
Ok,
see my
bueno, me
presentation?
presento, yo soy Rodrigo Astete, yo soy
I'm Rodrigo
especialista en sanidad
Astete,
agropecuaria y en cuidado del ambiente
specialist
de IICA.
in regional control of citrus breeding, science and management. And I will change a little view, the focus on the presentation, but we have been working with from IICA and innovation and surveillance. Our viewpoint is from the organization. So first, I would like to tell you who we are. Tell you about IICA. IICA is an agency that is specialized in agriculture of the Inter-American system, and we work with 34 member states for agricultural development and rural well-being. We are an institution that work on modern and we work with modern and efficient institutions to produce knowledge for competitive and inclusive agriculture. And especially SAIA is part of IICA's hemispheric programs. And what we work for to strengthen the health and safety capabilities. We try to implement international standards to improve response to phytosanitary emergencies. We also promote safe commercialization and trade, and we do networking with 50 specialists in 5 regions: North America, Central America, the Andean region, the Caribbean, and South America. I'm from Chile, but today I'm in Peru. What's the work of IICA? The Plant Health Committee, POSAVI, it's a regional intergovernmental organization, a regional one, that coordinates and joins efforts among different member countries that are Argentina, Bolivia, Brazil, Chile, Paraguay, Peru, and Uruguay, to address and solve common phytosanitary problems, to strengthen regional integration, and to protect agricultural production from pests and diseases. We have an agreement of cooperation, and this is part of our name, IICA COSAVE. It's because we have an agreement that is it's a framework for regional technical cooperation in plant and health. This has been approved by the COSAVE steering committee, and we try to create modern technologies. What we would like to tell you today is about a pilot between IICA and COSAVE that is a project, an initiative project for the early detection of stress symptomatology in citrus orchards. And we have 2 pilot projects to do early detection of HLB in citrus. We are working with our member countries in COSAVE.
Tanto de cámaras térmicas como hiperespectrales. Y obviamente responde a una problemática actual que tienen las organizaciones nacionales de protección fitosanitaria con la vigilancia convencional.
And this is in line with conventional surveillance, which is pretty limited. And of course, the idea is to move forward towards an early detection. The objective is to strengthen decision-making and what is the purview of every national body. This is the US entity, IICA in Mexico, SATIA in Chile, and SENASA in many countries. So our objective is to use high-resolution satellite imagery so we can orient. This also is in line with what Romina was saying. We can locate national organizations in an early detection so we can move forward with different analysis. such as the ones that Romina was mentioning.
Vamos a contar un poco de la historia.
Well,
Esto parte anteriormente con un piloto del 2022,
this
2023,
started with a pilot in 2022-2023 with a survey of base spectral signatures. This was conducted among INTA, COSABE, and IICA. It was conducted in the province of Corrientes in the northeast of Argentina. This was very successful, and we were able to separate positive and negative HLB plants. With these two pilots, what we are aiming for is to have the early detection, but we also worked on detecting biotic and abiotic diseases. The study will be conducted both in Argentina and in Uruguay. One of our consultants, Dr. Tejada from Spain, is working on hyperspectral indexes. This is a sample or a graph from Poblete in the year 2020. And this is the same we are doing with HLB. This is the early detection without symptoms with high resolution satellite imagery. These are both hyperspectral or thermal images. This started with an internship of experts comprised by Chilean and Argentinian scientists. And we wanted to come up with a joint work of technology transfer and early detection. This was conducted in the Pantalab lab in Spain, and we conducted a follow-up for 4 months through field activities to analyze the campaign with the hyperspectral chambers and with everything that was conducted in Argentina and Uruguay. Another picture, I think we're running out of time, so these are the activities that were conducted with drones and hyperspectral cameras or thermal. Then we have the use of head wall sun-induced fluorescence imaging sensors and different tools that were used. There was another pilot, which is the generation of heat maps for the targeted sampling. This is a citrus species grown in Argentina and Uruguay in the river area, in the northeastern area. This is so we could survey a prototype and then to move on to a second prototype in the whole surface. The idea is to come up with heat maps that can guide the direct work that could be done, for example, in this case, with the satellite or drones. And lastly, as Dr. Romina mentioned, through the application of PCR techniques and others to detect this disease. In terms of our expected outcomes, those are the strengthening of surveillance in the southern cone, technological innovation applied to HLB control, improved regional capacity to respond to the emerging pests. Of course, we want to achieve synergy between the member countries of IICA and COSABE. Some final reflections. Currently, national organizations conduct conventional surveillance that is very much on the reactive side. It depends on the visible appearance of symptoms or vectors. It's limited in terms of resources, and it's in an extensive growth area, 1 million hectares. So, of course, this creates a lot of logistical hurdles. It entails high costs. There is insufficient coverage, and there's a delay in detection. And this type of problems or pests is informed or reported by the grower, and not many times can we act in a proactive manner. So we are working on an early surveillance with technology, a proactive and preventative surveillance that can detect the appearance of biotics, abiotics, with the use of satellite imagery, hyperspectral thermal drones, with analysis algorithms, so we can have greater precision and coverage in less time. And we also want to optimize our resources and strengthen our technical capabilities. Of course, this started with a pilot project, but this is, of course, scalable to the rest of the region. The strategic relevance for the national organizations is that early detection, of course, reduces economic and phytosanitary losses. It strengthens decision-making, and it contributes to the protection of regional citriculture through rapid and coordinated actions. As a conclusion, the results of both pilots laid a groundwork for a regional early detection model designed to be scaled up to COSAVE countries and adapted to different agriculture production systems. And the final message, I try to be as brief as possible, but I want to convey that we need strategic alliances. IICA and COSAVE and the regional organizations in the Americas can partner with others. Here we are gathered with the University of Florida. We need to strengthen these alliances. FONTAGRO is a key ally in agriculture, financing, and innovation. We need to work in technological innovation. Of course, we need to advance in the use of drones, satellite images, and phytosanitary intelligence of a prospective type. We need tools for early detection and rapid response. And of course, we need to act together, strengthening national capacities. We need a regional coordination among governments, institutions, and academia. And we need to create more important projects under the early detection system of this particular pest, HLB. And of course, we believe that citriculture is not undermined. We don't want to put production at risk, and we want to come up with a more resilient and sustainable agriculture for the region. So this is the final message, and thank you. Thank you very much, Rodrigo. You have presented an overview of the southern cone of the Americas, and this is a strategy that fosters collaboration in the region. So we've had a pretty good panel number one. Now we will have a brief recess for 5 minutes, and we will be back at 10:30. And we will start our second panel. Thank you everyone for joining and staying with us.
Muy bien. Retomamos entonces la jornada. Muchas gracias
Okay,
por estar
so
aquí.
we will
A
continue now.
continuación, comenzamos con el panel 2.
Thank you so much. We will begin with
Empieza a trabajar
the
con el
second
tema de los
panel,
vectores, algo ya aparecieron los
which
vectores
will start
en la parte de
addressing
diagnóstico, pero
vectors in
ahora vamos a
diagnosis.
tratarlo
We first
específicamente,
had
sobre
the
todo el manejo
diagnosis part.
integrado,
Now
a
we're
partir
going
de
to
dos
speak about
presentaciones. La primera
comprehensive
estará a cargo
management.
de
The first
Chuck
presentation
Niblett.
will be in charge
Chuck,
of Chuck
¿estás por ahí
Nivet.
para hacer tu presentación?
Chuck, are you there?
Yes, you can talk.
Yes. How do we turn
Muy
me up?
bien. Thank you very
Yes,
much.
here we are. Well, thank you so much.
There you go.
I'm able to start video.
I guess
We
we
can
need
hear you.
Yeah. Do I need
We
some
can
help
hear
from
you
Martin,
both.
to turn on my video?
Yeah.
Yeah.
Yes. Yes, because we are unable to start.
Oh,
The
we are
host
unable.
needs to
We are unable to start the video.
It's okay.
The host needs to enable it.
Let me write in the chat. Oh yeah, start there. Now.
There you go. Now we can see you. Now we need you
Thank
to
you.
share the presentation.
And this is Anna. As you'll see, she's also a
Hi, Anna.
plant pathologist. And then I
Hi.
have my angel Angie, who got my computer set up to work.
This is our
Great.
video and audio producer. I'm the other part of Venganza.
Okay.
Okay.
Now start the video.
Okay.
One of the slides.
You need to share your screen.
Can you see them?
Not yet.
Oh, okay. So let me go back. Sorry, I need to then do it by sharing here. Share. Yeah, Microsoft PowerPoint. Yes. No, share. Can you
There
see it
you
now?
go. Yes.
Okay.
Now you full presentation. Perfect.
Okay.
We're all set then.
Okay. The title is a little different than what is advertised. I'm going to talk about some work that Anna and I and a bunch of colleagues have done over the last 20 years, applying RNAi or RNA interference or gene silencing to controlling several different types of plant pests. And this is a little about our backgrounds. As you can see, we both have PhDs from UC Riverside. I was the department chairman and professor of plant pathology at UF for about 21 years. And listening to Rick's presentation was really heartening because I've been away from UF and their research for, as I say, about 20, well, however, since 2002. And the amount of work that's going on citrus and on many other crops is really inspiring. During that time, Anna and I developed Venganza, and some of the work that you'll see reflects that. Anna is a native Mexican. She was a professor at Sinvastav in Irapuato for 18 years, and Sinvastav is probably the premier plant science research in Mexico. We founded Venganza in 2002. And I would say at the outset that these slides are freely available. If anyone would like a copy of them, all they have to do is ask. We're happy to share them. And also later during the question answer period, since so many of your participants are Spanish speakers, why Ana can answer any of the questions in either English or Spanish. So why do we do what we do? We do it to try to solve a global problem because pathogens and insects cause major losses worldwide. A lot of the controls are chemical. You have the purchase costs and the application costs, the toxicity and environmental contamination, and also, as Rick suggested, for the antibiotics, the pests can develop resistance to them. So our approach offers a new opportunity for plant breeders because the genes that we get from the fungus, actually, the fungus we work with mostly, are actually genes that the plant breeders can use in breeding subsequent crops. And the important thing about transgenes is that they are simply inherited as single dominant genes, which is every plant breeder's dream. So this is what we started with, the black shank of tobacco, because Anna had a lot of experience with Phytophthora. And this is basically what we do. As most of you know, venganza is the Spanish word for revenge, and you'll see why later. What we do is to identify an essential gene in the pathogen or the pest that it needs to live, to reproduce, cause disease, or even to produce mycotoxins. We then take that gene and put it into a plant transformation vector that produces double-stranded RNAs. Double-stranded RNAs have no role in plants, so the plant immediately recognizes it as foreign, and its nuclease enzyme dicer chops up the invading double-stranded RNA into small pieces called small interfering RNAs, or siRNAs. Then when the fungus or the insect feed on the plant, they take up these small pieces, and then their enzyme, the dicer, silences that critical gene in the pest. So it inactivates or kills the fungus or insect, the plant is resistant. So the plant, we say, gets revenge on the pest using the pest's own genes, hence the name Venganza. So as I said before, in addition, this approach provides to plant breeders a whole new series of genes for resistance. The genes of the pests themselves now provide the source of resistance. This is a little prettier description of how it works. I won't spend a lot of time on this, except up in the upper left, you'll see we have a transgenic plant transformed by that structure up there under number 1. The plant translates that into an RNA. The RNA folds over into that double-stranded hairpin structure that I talked about, is recognized as foreign in red letters by the plant. The RISC, the RNA-induced silencing complex, which are enzymes in the plant, pick up those little pieces or attach to the RNA, cut it up in the little 19 to 23 base pair siRNAs. All this happens inside the plant. You can see down at the bottom there where it says soil, the orange eggs represent the pathogen. So this is what happens in the plant. Then when the pathogen penetrates the plant, we get to number 3, it picks up the siRNAs, and then the fungal gene is transcribed to messenger RNA, and the messenger RNA is cleaved. So there's no gene expression in the fungus, and the fungus either is greatly retarded in its development or dies. This was the first product that we produced. You can see the black shank on the wild type or susceptible plant, and in the foreground, the resistant plant. This shows you how it really works. On the left panel, you see that we isolated the RNAs from the fungus, and the wild type culture produces the 620 nucleotide message, and the transgenic cultures, which are transformed with the fungus, with the DNA construct, they're all chopped up and show them under A, B, C, and D. The right panel shows the same thing with the 35 nucleotide control. A and B are plants that are wild type, there's no spot there, but then our transgenic plants, 4, 23, 26, and 27, all show the small interfering RNAs were produced. So this is kind of the, if you will, chemical proof that silencing is occurring, both in the fungus and also in the plant. And this was a fortuitous thing, an interesting observation. At the time we were doing this, there were a lot of tobacco plants in the greenhouse, and a lot of them were dying, except ours were not. The ones that we had made resistant to Phytophthora were also resistant to Peronospora, the blue mold fungus. So cross-species resistance is possible when the pests have similar gene sequences in their essential genes. Remember here now, we're using an essential gene of the fungus. This is what happens to the fungus. It really makes its life miserable. A is a normal fungus for those nice, healthy hyphae or mycelium, and then A through F show different treatments where they're silenced. They've been transformed with some of our constructs. So the fungus really, Anna's really a good mycologist, and she cries every time she looks at those poor sad fungi. And here's another example of it. This work was done in collaboration with some Australians. We made the DNA constructs and sent them. They transformed Arabidopsis and then treated the soil with the fungus Phytophthora cinnamomum. And you can see the results. The plants with gene A and gene B are quite large, whereas the wild type in the middle is badly affected by the Phytophthora cinnamomum. Now, if you were growing a crop of Arabidopsis or any other plant, you'd certainly want the ones that are treated with gene A and B and not the wild type. Down below, you can see the big spots there from the where we isolated the siRNAs. And then the blue vertical bars show you that the length of the roots was much greater in the transgenic A and B than in the wild type. Anna developed a lot of different methods to measure the effectiveness, to test the effectiveness of some of the different double-stranded RNAs that we were identifying and producing. You can see again on the top figure there with no dsRNA why the fungus looks pretty healthy, and then gene A, gene B, it really damages the fungus. And down below in the orange petri dishes, you can see the amount of radial growth in the control in the Cox plate, the elicitin plate. Very little growth in gene B and no growth in gene A. So this shows you how we test these molecules to see if they're going to be effective against the pests that we're targeting. Here's another method that she developed. This is for Fusarium, which attacks a lot of different plants, including citrus. With no double-stranded RNA on the left, all those black spots represent all of the germination of all the spores. If we try ribosomal RNA, you can see we knocked it down to maybe a half, and then gene X knocks it down to maybe 20, 25%. We use ribosomal RNA there in the middle as a control, ribosomal RNA from the particular host that we're targeting, because there are multiple sequences of it and in the genome, and we feel if we can knock that down, then we can knock down any single genes as well. So that's kind of our scheme. We use ribosomal RNA as a baseline. And here's another example of a fungus that can't be cultured, Puccinia graminis. It causes stem rust of wheat. You can see in the upper left, the control, it looks like a bunch of spaghetti. And then when we treat it with different double-stranded RNAs, you can see the great reductions, 80%, 60%, and 99% by some of the dsRNAs. So these are the methods that we use. Now, in addition to the work that we've done, mostly with the fungi, we've collaborated with several other investigators in different parts of the world. And this is some work we did with the people at SIP in Peru, and also Ghent University. The work was done primarily by Catherine Prentice, working on the sweet potato weevil. And Catherine can cause RNAi to work either by injecting the double-stranded RNAs into the pest or by feeding it. Here you can see the effects of injecting a sequence from the lacase gene, which causes the coloration in the sweet potato weevil. In the left column, you can see the colored ones, the dark ones, and the pupa. And then in the right column, you can see after several weeks, probably, or days at least, of lacase treatment, that they're essentially albinos or colorless. The lacase gene has been silenced. And these vertical lines show the effect of different double-stranded RNA sequences on the sweet potato weevil. The taller the line, the better, because that shows 100% mortality. In the lower left, you can see the control. That's probably normal mortality during the time period that the insects were kept. And again, these were injected. So you can see that there are many different genes that we can target by this mechanism to control this insect. And so again, this is by injection. These figures show the effect of feeding. And again, the upper lines show the mortality of 2 or 3 of the different double-stranded RNAs. The lower dotted line is the control, and this simply shows different concentrations, so it is concentration dependent, but the effect is still there for those particular genes. This is some work we did with people in Uganda with Bioversity and the Uganda National Agricultural Research program. It was done by Walter Osamati, who was a student there at NARO. And he came to our lab for 6 weeks when we were in Raleigh, and he did most of this work himself. And he and Anna selected the sequences to test against the banana weevil. And then he took those home with him and developed an assay and was able to treat the larvae. And here you can see the effect of gene in plate A, the effect of gene 20. You can see those little dots there. That's how large the larvae grow after being treated with gene 20 as compared to the water control. And gene 11 is over there in plate B, again, in comparison with the water treatment. Here, he's just showing it a different way. His name was Walter, and so up there under gene ID, he's got Walter rather than water. It should be water control. But you can see at the different levels that he gets very good reduction in growth rate of the banana weevil using the UBE, the ubiquitin gene, as compared to the ribosomal RNA. So very good control. And again, down below, you can see the little ones that were in the ubiquitin double-stranded RNA compared to the large ones that were in water. And of course, everyone knows the psyllid. And some work has been done with it. This work was done by Wayne Hunter, USDA, down at Fort Pierce. This is the system that they use to keep the citrus leaves green while they fed the psyllid on them. It's basically a couple of magenta jars together with a piece of styrofoam and a microfuge tube, and the citrus will stay alive for a couple, 3 weeks in there. And then on the far right, you can see the dead psyllids on the top of the styrofoam. So the psyllid double-strand, gene silencing works against the psyllids in citrus. There'll be some questions about trying to control HLB with RNAi and also with the psyllid, because neither of those species have the dicer genes in their genome. But in this case, Wayne has shown that taking up the double-stranded RNA by the citrus does kill the psyllids. So, and we're wanting to show the same thing with the HLB. It lives there quite richly in the phloem of citrus trees. And so if we introduce the gene silencing constructs there, they will essentially be bathed in it in the phloem and should be, their growth should be slowed down or killed. So this is the psyllid. Again, Wayne, some of Wayne's work, you can see the 2 higher lines there show the effect of 2 of his double-stranded RNAs in comparison to the others down below. And then the gray vertical lines down below actually show the levels of gene expression with it reduced by the 2 double-stranded RNA molecules as compared to the controls. This is some work that Lucas Stalinsky did. There are 6 different boxes there, If you look at the upper left one, the tall lines on the left are his controls over with different concentrations of the controls. The three on the right, the three lines on the right that diminish as you go to the right, those are the treatments. And you can see that in the three boxes on the left, he got control with all of those genes. Same, the two upper boxes on the right, and then the bottom box down on the bottom, that shows that the control molecule did not have an effect. So this shows that the RNAi is alive and well in the psyllid, even though it does not contain the dicer gene sequences in its genome. And then another serious pest in agriculture, the white flies. There are two major species of it, and they cause damage to the plants. They're toxins, they transmit numerous viruses, and they are susceptible both to RNAi. We're doing some work with that with Judy Brown at the University of Arizona. She has quite a long reputation of working with the white flies. So this is our solution. It's a proprietary RNAi technology to control the pests. Our transgenic plants will reduce and limit plant pest epidemics, reduce pesticide applications and contamination, be less likely to create pesticide-resistant strains of pests, and again, provide new genes for pest resistance to plant breeders. I'd like to go back to number 3 there because that's important because, again, as Rick mentioned, we always are concerned that pests will develop resistance to the treatment that we're trying to use against them. In this particular case, it would be very difficult for the pathogen to develop resistance to RNAi because we're using its own gene. It would have to mutate that gene considerably within its own genome in order to overcome the resistance to the sequence of the gene that we're using. So there's a high probability that this type of resistance would be very long-lived. The other thing is that when you make the DNA inducing constructs, you can stack different genes together on the same construct so that you can target multiple genes in a single pest. You can target multiple pests with all in the same construct. So there's a great latitude and facility there with RNAi. So we think that there is a bright future for RNAi as part of the transgenic plant approach to controlling plant pests and diseases. It's one barrier that we will have to overcome, the use of transgenic or GM plants in our society. But I think with something like as important as HLB and some of the other diseases, that this resistance to GMOs will become much less in the future. So thank you very much. We're happy to entertain your questions at that time. Go ahead. Yeah.
Thank you, Chuck and Anna. Wonderful presentation and an extraordinary strategy that apparently works, not apparently, it works with under many circumstances with different organisms. Now, well, Lucas is already sharing his screen, so we welcome Lucas for his talk.
Good morning. I'm going to give a kind of a brief history. It's easy to talk about, I guess what I'm calling here mistakes in hindsight, but I'll try to focus more on the opportunities. So just to remind everyone what happened in Florida, the psyllid was found, the vector in 1998, and the pathogen was confirmed several years later. So there's a period I call squabbling, I guess, research over determining diagnostics. Again, in hindsight, it's easy to be a critic. Probably would have some more urgency at that time may have benefited us, but that's what it takes in science. Sometimes it takes a long period of time. I believe personally that we had the pathogen and the vector in 1998, at least at in some level of pathogen. So you can see that several years of allowing this pathogen to spread potentially where nothing was done. And really, vector management didn't become accepted statewide as necessary until about 2010. And that had a lot to do with the latent period of, there's a long latent period before you see symptoms. And really there were, at that time, we put the brakes on research ourselves. There were kind of silly regulations like this pathogen was on the select agent list, which hampered research to some point. So 12 years essentially elapsed before the vector was being controlled. And by that time, the disease was probably well spread throughout the state. And one of the critical things of management of this disease, inoculum removal, really never gained ground. And I think Michael Rogers will talk about that tomorrow. This has a large sociological component. This was probably in large part the outcome of several previous attempts to eradicate citrus canker, or simply didn't want to remove their trees at that point. When we did begin to control the vector in 2010, it became quite intense, quite quickly. Although this is you could see a typical spray regime, just one example, but there were, for those who are managing between 10 and 18, this is kind of what it looked like. It was essentially monthly. This was expensive, and this eliminated natural enemies, which played a large role in citrus pest management. That was really a great example of biological control in the past. Well, growers were implementing this management strategy, and greening was showing up anyway because it was probably already in their groves once they began to manage the vector intensely. So the typical model for sprays during that time was to put out a dormant spray application after harvest when the new flush was available for the vector. And that was really a weak link in their life cycle where you could really get a good start on the season. And then sprays thereafter were just made on a calendar schedule where the intervals between sprays were determined by length of efficacy. So this was really a traditional kind of pesticide treadmill, which quickly resulted in insecticide resistance to a number of pesticides, which we did get a handle on eventually through effective rotation. Nonetheless, populations in Florida remain quite primed for resistance. This is an example of an experiment where I went into a location which had heavy neonicotinoid resistance when we came in and prior to implementing an effective rotation. And then we got the population to be susceptible through that rotation after about a period of 6 months. And then I put in an experiment where I didn't rotate neonics for 3 consecutive sprays, and I had two types of rotations, A and B, or 2 different types of rescue rotations, as I call them. And this, on the y-axis, is the resistance ratio. It essentially tells you how resistant the population is. The higher, the worse. And after 3 consecutive applications of a neonicotinoid in this location, you quickly rise. You get a population that's 2,000-fold less susceptible to that neonicotinoid. And if you look at the actual psyllid catches in that non-rotated plot versus the 2 rotations, you can see that we lose control of the psyllids. And when we put the rescue rotation back in, we can knock the psyllids back. So this resistance that we see is absolutely coincident with failure to control the vector. By 2010 or 18 rather, kind of right before that falling off the cliff that Rick was talking about, growers began to question the need for controlling the vector at all. You know, if all of our trees are infected, why control the vector? We actually got some funding from USDA MAC at that time to look at this question specifically. And this was so far into the disease problem in Florida, we still at that point didn't have answers to these really basic questions. So this is an example of what trees look like a year after I began to challenge them with various inoculation frequencies of vector with and without pathogen. And the top panel will show you vector plus the pathogen, and the bottom panel only shows you non-infected vectors. There's no pathogen there. So a year after a single infection, this is what trees look like. On top, this is a tree with a year of HLB, and the bottom one just had vectors on it for a week a year ago. So you can see the tree with HLB is less robust than its counterpart, essentially no pathogen control below. You can kind of see through that tree, and it's yellow. Here are trees that receive 12 inoculations. So this is like a monthly grower spray. And you can see that with 12 releases of infected psyllids and then killing those populations off, the tree doesn't look very different. Those trees don't look very different than the ones that just have HLB and no psyllids for a year. And if you don't do anything at all, the trees collapse. So this got us to thinking, you know, the trees can survive. Basically, what this says is, yeah, you do need to control the vector, even though all your trees are infected. Even though we're 100% infected in Florida, you still have to do something about the vectors if you want your trees to look like this rather than completely collapsed. And you can see that these trees with 12 sprays don't look very different than trees that just were inoculated once. So it got us to thinking that there might be a threshold with which we could survive, below which if we could keep psyllids below that threshold, we could keep the trees alive and without the need for continuous spraying. So this is what this concept would look like. You would still spray in the wintertime when the life cycle is at its sort of a key time where you can knock down the populations. And you can keep that on a calendar basis, but then monitor psyllids. And once they reach this threshold where they're contributing to the decline of the tree, you got to knock them back. And perhaps you don't have to that way spray on a calendar every month, because growers can no longer afford to spray 12 times per year with the yields that they were getting. Key assumptions here is that your goal is not to prevent spread of HLB. It's just to keep the trees alive. And in fact, you can experimentally verify this. This is going into the same plot that has all the trees are infected with HLB, have the same level of infection. And you only treat for the control during the overwintering period. The calendar gets 12 sprays a year, and these 0.2 psyllids per tap and 0.7 psyllids per tap are two thresholds that indicate when we would spray. And what these data shows the cumulative Asian citrus psyllid catch over that year. And what you see is the more times you spray, the lower your psyllid population is. And it turns out the more times you spray, the higher your yield is. So it in fact does contribute to increasing yield if you control the psyllids. But we got equivalent profits between 4 sprays and the additional 10 sprays, meaning at some point, if you're just on a calendar, you're just throwing your money away too. You're overspraying. So these thresholds can be a useful way to decrease the insecticide input and still keep your trees alive. despite HLB. And if you don't spray at all, your trees are going to collapse, like I showed in that picture. So we have quite a large toolbox now of psyllid management options. Of course, tolerance and resistance that Rick talked about with the breeding and RNAi, that's really coming online now. And those things are going to be so exciting once they, it takes a long time, as Rick mentioned, for those things to bear fruit, so to speak, pardon the pun. But they are now. Of course, we've been really focusing on removal of vectors with insecticides, trap crops, repellents, natural enemies. And none of these things work alone. You're going to have to put all these things together really to control HLB. And we have a lot of those tools that are showing really good effectiveness. You can repel psyllids and decrease landing and egg laying with repellent mulches. This is not going to cure HLB, but it could be part of an integrated management program. These tree defender bags, really, they exclude the vectors, get trees up and running before several years. They give them a several year head start before the bags are taken off, so we could get trees that yield. Kale and clay, which deters and repels psyllid egg laying. Of course, growing citrus completely under cover structures. All of these things have proven quite highly effective. Now, the next generation tools have come, and during that period, while we've been waiting for those, antibiotics have played a prominent role as a stopgap. Initially, they were tested by spraying, and unfortunately, spraying these didn't work. We couldn't get enough antibiotic into the tree where it was necessary. But this didn't deter researchers from saying, hey, all right, spraying didn't work. Now let's start injecting these things. So very quickly after determining that the antibiotic sprays were not going to work, there were labels for injectable antibiotics after data showed that they showed promise. And in fact, these are different formulations of Oxytet. When you inject them into trees, you find quite long-lasting decreases in the count of bacteria in the phloem, even though the antibiotics don't last in the phloem this long. It just takes the bacteria much longer to kind of regrow. And you can see a control tree here versus an injected tree after 2 years. The difference was quite spectacular for us in these early tests. These were tests when we were injecting twice a year in this particular experiment before those formulations had been labeled. And we can see a yield improvement. And the thing about these data is these results have now been replicated in the hands of many researchers. And when I see the same results coming out of multiple laboratories, that's when I really start to gain confidence for something. This has really been in my mind, a game-changing stopgap. Opportunities for the future, as Chuck mentioned, transgenic technologies offer one possible opportunity. Working in a team led by Brian E. Bonning, we've transformed sweet orange, grapefruit, as well as the citrus relative with 2 BTs that we in earlier experiments determined were effective in killing Asian citrus psyllid, particularly the early instars. These trees will reduce adult populations, and they severely impact emergence of F1s. Our initial idea was maybe we could use these as trap plants with the citrus relatives, because knowing what a hurdle will be to overcome regulatory acceptance of a transgenic tree, but maybe transgenic trees aren't a bridge too far now. We're starting to think, and what you can see these BTs. This is a gut of a transect of a gut of a psyllid taken from a wild type citrus tree versus the transgenic one expressing the BT protein. The BT protein punctures holes in the gut of the psyllid and kills it in that manner. And there are a host of other technologies now coming online. As Rick mentioned, I'm also particularly excited about progress towards potential HLB tolerant lines that express a more effective immune response, these NPR1 trees that Rick mentioned. There are other potential possibilities with genetic engineering, including getting various antimicrobial peptides into the trees, perhaps BT against the vector. And threshold guided sprays should allow more tolerant trees to flourish, right? Tolerance would simply allow for higher treatment thresholds and fewer sprays. So I could see how these technologies would simply, and practices would work hand in hand in a new management system as these things are put out in the field. Chuck was mentioning RNAi. These are just the examples of RNAi discovery with Asian citrus psyllid that I've been involved in. And I know there are many others because I haven't. There's other ones that Chuck even mentioned. So CYP genes have been targeted with RNAi in Asian citrus psyllid. This has been done with oral and feeding solutions, and this increases their susceptibility to insecticides. Cathepsin's B and L have been effectively silenced with virus-induced gene silencing. So these dsRNAs were put onto a virus that was then the tree was infected with. And this reduces ovarial development and reproduction in Asian citrus psyllid. Nabil Killeney developed a super neat triple construct with 3 dsRNAs attached to a CTV virus, where when the plants become infected with this, it changes the way the plants look and smell so as to attract psyllids. And then those attracted psyllids that feed on the plant have reduced fecundity and fertility, which is really neat. Recently, this is in review now with the team I've been working with. We've silenced gut surface proteins on the psyllid gut, which reduces acquisition and transmission of the pathogen. And another project that we've been working on, gosh, how do I go back? Knocks down this vacuole sorting protein, and this reduces fecundity, fertility, kills the psyllids. So the discovery has been done. Now we need products to come out of this. Last year, I don't remember what the scientific breakthrough of the year was that was named by Science Magazine, but I do remember that the runner-up was the first RNAi insecticide released against Colorado potato beetle. So there's already a commercial RNAi insecticide that's been commercially released. So now we need somebody to take this discovery and get these out there so they can be used. So I only have a short-term outlook because we need answers, and now antibiotics are a promising stopgap. I firmly believe that. I think we can manage the vector at reduced cost with thresholds. We, as Rick mentioned, we can minimize fruit drop with this immunotherapy with using hormones. That's really proving to be quite effective. And the time for these next generation tools is now. I don't think there's any more, as researchers, we can't say 5 years from now, 10 years from now. We need those tools now. So that's all I have. Thanks.
Thank you, Lucas, for your talk, and providing with this urgency for action. So we have finished panel 2, and now we have some minutes with Charlie, and is Eugenia there?
It is.
Okay.
Hello.
Welcome, Eugenia.
Well, thank you very much, Martin. I really want to say thank you to all the panelists today, and a special thank to you, Martin, and FONTAGRO team, and also the team in the University of Florida, as well as Charlie. The presentation had been great. I'm really interested in see how we together can start identifying all the gaps that we have in knowledge and how together we can accomplish on the next generation of the solution that we can bring to the industrial sector of the citrus. I don't know if everyone wanna turn on in the camera, because I think all of you have said a lot of very important information, not only for maybe the people that is right now attending virtually in the panel, but for sure many other will attend or will see the video later, as always happen. I don't see Charlie as well. Oh, Charlie, you are there, yeah. So I would like to say that this citrus screenings is one of the more destructive disease that we have in the industry. It have been here for a long time. FONTAGRO have been working on this also since 2017. But I think we have the opportunity now to start getting together all of you and also all the region, the Latin American regions through us, and see how we can address an initiative that can help the next generation of technological solutions, or how we can work better with farmers and early detection systems and many other, ideas that you can bring to this new platform. So I just want to say thank you. We are looking forward to work together for sure. And I would like to pass the mic to Charlie.
Thank you, Eugenia. It was a fantastic session. I think it's a great opportunity. I think we need to get our heads beyond the orchard and start thinking at this hemispheric scale. There's a lot of exciting methods. We need to think about how you're going to scale it. But there's a lot of food for thought here. A couple of 3 themes that come to mind, maybe distill all this, is the detection, the IICA presentation, and Romina and Rick. It tells me we have some of the tools, things are brewing, but we need to think about how we're going to do all this detection asymptomatic. We cannot fly in drones in every single orchard, or maybe we can, not sure about Florida with some restrictions, but can we use satellite? How can we leverage that satellite information a little bit better? And how that system comes into place at this sort of hemispheric scale. Resistance is the other one that came in all the talks in different ways. We need to understand how we are going to manage the resistance. I think the evidence from the insects with the insecticides is quite humbling. The same thing will happen to BT, and is RNAi part of the solution, or stacks? I think there's a tremendous area of research that I don't think has been done yet, and we should do a little bit more in there. And stacks and regulatory, hopefully we'll learn from Mercedes tomorrow more about the regulatory systems to deploy the stacks. The more things you put in a cassette, the more hurdles you need to go through the regulatory. So I think with these 3 themes in mind, I hope we can have a conversation, but not thinking about the tree or the orchard, but at the whole region, right? So yeah, we can fly a drone, we can take pictures, we can look at the oranges in one place, but how we do the detection, prediction, asymptomatic at the continental scale, right? How we are going to contain this disease and eradicate it, hopefully. So I will pass to Martin and hopefully get him to comment about and what are his thoughts.
Oh, thank you, Charlie. I was amazed by the battery of solutions that all the presentations managed to show us. A lot of good research going on, attacking different parts of the problem and finding some very promising solutions, some effective solutions or proven solutions. As Charlie mentioned, the talk by Rodrigo and the predisposition of everyone here to collaborate at the regional level is very promising because we have a sort of lab setting at the regional level with real needs and we can at the Latin America, Caribbean, and North America level find some solutions to this problem through collaboration. While I was listening to the talks, I was thinking of some sort of lacking integration of these different strategies and how we could recognize them in terms of TRLs, technology readiness level. Some people are working on basic stuff, some people are working on applied stuff, some people are developing, or the last talk was almost pride, we need products. And I think that just mapping the solutions that we have in terms of a matrix with TRL level will help us to find where we are at and how we can integrate these different approaches and come up with something close to solutions. That's my humble contribution.
Thank you, Martin. I think the integrated approach is what we need to follow, and I think all of us or all of yours that you are working on this topic for a long time, as well as many others, will help to identify those gaps that we need to start implementing through the whole hemisphere. We have had in FONTAGRO other experiences similar with Fusarium in bananas and plantain, and we have to take a similar approach, trying to bring all the knowledge that we have for technologies or also knowledge that help farmers in the short term, but also we need to think in the medium and long term. In that view, I would like to have some ideas from all of you in this closing section of the webinar. So I would like to invite you, all of you, to maybe express a comment, an idea, or what do you think, what we need to focus next.
Maybe since everybody's quiet, I will ask Rodrigo how would he envision a project to scale his research to Florida to the south, to Argentina? Is that possible? What would you think about that strategy?
Unfortunately, Rodrigo had to leave us before the closing session.
But I would like to start for Dr. Stelinsky. That was your last presentation, and I like one of the slides that you present, that we have a large toolbox for ACP management. What do you think is the next steps for an integrated approach for the whole hemisphere?
Well, I like the idea of that Martin suggested of maybe doing some kind of matrix of what we have and what is coming online. I think that through what we have a craft program in Florida that reimburses growers for allowing people to come in, researchers to come in and test their ideas for integrated management programs. are getting at what I was suggesting. I think really this is the way of going into it. And I see Michelle raised her hand where I know they have a grove where they do this. We just need more of this, of integrating multiple tools. We have things that work quite well, and the problem is that this is an ever-changing target, and as the industry in Florida declines more and more, the tools that we have now become more and more out of reach, because you need to control the pathogen somehow with an antibiotic. You have to control the vector. You have to feed the tree. You have to, and growers just don't have the money for all those. So we have to have a sliding scale of what is the minimum that you need to do, and then prioritize where do you put the investment in. All of this has to be done within an economic framework of where do you get the most benefit out of the investment. So these things have to be on a sliding scale, or they have to be target specific. They can't be broad spectrum, one size fits all. It's a complicated answer to a very hard question.
Thank you very much. I agree, because most of the farmers, they don't implement just one tool or one technological solution. What they do is just combine different solutions, and that has an economic impact at the farm level as well. So I will pass the mic to Michelle.
Yes, thanks a lot for giving me the opportunity to weigh in here. I would have to agree that designated test sites to perform research on integration of all of these strategies is the highest priority. The challenge is that many of these technologies are not approved for use in commercial production by the U.S. Environmental Protection Agency and APHIS and the FDA. So the trial sites have to be managed as research sites. And that means, reimbursing the participating growers for their cooperation at a very different level than, reimbursement for just the, crop destruct. When you start putting experimental treatments out into the grove, every block of trees receiving a treatment starts to respond differently and needs to be managed very uniquely. And that has to be taken into consideration. As well, you have these perimeter and isolation zones around the trial sites that need to be maintained until data can be collected on the impact of some of these technologies on the environment. So it is complex, but it is doable. And I think if we are motivated to come together as a group of researchers to establish this, it can happen. And there are opportunities to do this with funding now through programs in APHIS like HLB MAC. And we have a new grant that will allow us to do this for the first time in a 20-acre field trial site in Florida to integrate across a few different technologies. And so I think we just need more of this. And we can get to some answers where, which of the technologies can come together and work synergistically and need to be, advanced further in the laboratory.
Thank you very much, Michelle. Now, Maria Mercedes.
Yes, thank you. Well, it's been an amazing discussion, and I've certainly learned a lot. And it brought me back to my years of working with Judy Brown on RNAi tomato in Honduras, and then working on a lethal yellowing project for 12 years, where I couldn't do a thing. I'll tell you a little bit more tomorrow. But I take courage from what Chuck says, the tree of the future. I totally agree that we need to work on that. And what Michelle just said, it's complex, but it can be done. And let me remind you that Roger Beachy and Dennis Gonsalves got their transgenic plum and their transgenic papaya 25, 30 years ago. Francisco Aragao in Brazil commercializes already the GM bean with RNAi technology. The BT has been difficult. And Charlie, I don't think we have an easy ride at all. I know that, we know that. And yet it is the way forward because it is the way forward. And let me tell you from my time in lethal yellowing research, this coconut disease that is a phytoplasm is very, very similar to HLB. Using antibiotics really scares me. I invite everyone to think of the One Health approach. So the One Health recognizes that human, animal, plant, and ecosystem disease is all interrelated. I can't think of spraying antibiotics on trees, which we had doesn't work. And injecting every tree with antibiotics and then eating those oranges, what it does to the soil microbiota. I don't think that's the way forward. That's a very personal thing, and I invite everyone to think of the One Health approach. So we have no choice, Charlie, everyone, but to plunge ahead. And as I guess it was Einstein who said this, that doing the same thing and expecting different results is just madness. We've done something very wrong with the GM approach, managing the GM activism. It's still there, it's still alive, it's still very strong, as you all know, with your esteemed colleague Kevin Folter that should participate in this. He is the champion of knowing what we're fighting against. But we've got to do it, we've got to succeed because we have no other choice. Thank you.
Thank you very much, Maria Mercedes. Fred, I see your hands raised.
Yes, thank you. I want to touch a little bit on the Tree of the Future and something that Mr. Dantzler said earlier about the desire to fund a trial with the best rootstocks that have been produced by conventional breeding, along with the best Scion cultivars that have been produced by traditional breeding. Tomorrow, I hope to convince you that some of these have some very real high degree tolerance of HLB as a disease. And these are things, as we're talking about looking at multiple technologies together in large trials, that at least at the minimum, the genetic basis of the plants that we're planting ought to be considered as well, and to try to get some of these new advanced releases and experimental materials into some of these trials. Stay tuned for tomorrow. I have some photos of trees 7, 8 years old that you will look at and you will say, what is citrus greening? Without antibiotics, without the IPCs, just standard care. So that's what I wanted to add at this point. Include new genetic materials as we begin to look at multiple technologies as the solution to HLB. Thank you.
Thank you, Fred. So, Carrie, the floor is yours.
Thank you. Mine's a very quick one, and I also put it in the chat just so we'd have it. I, of course, come from a detection technology side, backing up Romina. She and I work in similar veins. While we're talking about how to manage, how to treat, I want to make sure that we are also targeting knowing where the organism is, so that as we're doing parallel tracks, right? None of this can be done in silo. The whole point of this is that we're doing coordinated and all fronts forward. We should not be forgetting that you have to actually know where this is. And Romina's presentation was pretty good about showing you can't just go by symptoms alone, because this silly thing looks just like nutrient deficiency, in part because the tree is expressing that it's hungry, because it's not getting the nutrition it needs to where it needs to go in the leaves. So I want to just note that we do need to know where it is and where your hot spots are, not just for the trials, because of course you want to put the trials where you have hot spots, but also for managing while the growers are struggling to survive now and waiting for the tree of the future, which maybe we're right on the precipice of, and I hope that is true. I would like to be hopeful and optimistic. So I just want to note that we don't want to leave behind detection. One of the struggles we've often had with detection of organisms on an international scale is not just the detection capability, and this one is super annoying because there are not lateral flow devices that are easy. It's got to go to PCR. So this is not a field friendly. So we could use detecting centers, spread accessibly, potentially, that need quick turnaround, and then the release of that data, real-time knowledge of where the organism is moving and where it's hot and where it's not, would be very useful for targeting the limited mitigation strategies we have at the moment, and then the deployment of testing sites to those places where we want to be able to really send these trees of the future through a rigorous environmental testing area. Thank you. I really enjoyed these conversations.
Thank you, Carrie. And you mentioned something that's very important and is related with how we can monitoring, the movement of the insect, and we have a webinar 2 weeks ago where that topic came in the webinar as well. So we need to rely more and more in all the ICT technologies and communication and information technologies, and see how we can use them to help all the farmers and the industry to detect and early detect any symptom or any hotspot that is related with this disease. So I would like to pass the microphone to Rick and then Romina, and then we are gonna do a closing with Charlie and Martin. Thank you.
Thank you. When I try to enable the video, it says you cannot start your video because it is disabled by the host. Not that I need to be seen, but I am still here.
Well, let's see the
I've
people
just
that
tried
is
to I've just tried to fix that.
okay.
Sorry, Rick.
But that's no problem. There we go. These have all been fair comments and good comments. But let me share with you what has been probably the biggest frustration that I have experienced in the 6 years I've been in this job. It is that it does not seem like anyone is trying to fashion a master plan to bring all of this together to lead to a solution. And maybe it's just the nature of how it works, that everybody's doing their own thing and each discipline is chugging away. And I'm sure there's more collaboration between researchers than it seems. But it just doesn't seem like anybody is taking the results from all of the projects that are funded and trying to identify something that could lead to a solution. That many of these final reports just end up on the shelf and that's the end of it. Here's an example. I happened to read that in 2018, Dr. Nian Wang found out that the combination of oxytetracycline and streptomycin together killed all of the pathogen. I mean, it was gone. I discovered that on my own maybe a year ago. And I was here in 2018. It was funded by a different program, so I didn't know anything about it. But at that point, I wish that someone had been looking at Dr. Wong's final report and come to my office and say, hey, we've got a potential solution here. And just think of all the carnage that has happened in the Florida citrus industry since 2018. But it just doesn't seem like anybody is bringing to the funders some sort of a master plan. The reason that we got excited about the Crop Transformation Center is that we saw that maybe there could be someone, i.e. Charlie Messina, that was taking everything that all of the molecular scientists were doing from a transgenic breeding approach and sort through all that to bring to the funders out the most likely success. We can't fund everything, and everybody's project has merit, or everybody's proposal has merit. But we just would hear about some great trees that had been created by this molecular scientist back in 2016, and then another one in 2018, but nothing was coming together. And so, we were hoping that the Crop Transformation Center, and I don't want to call it chaos because it wasn't that, but we were hoping that someone, i.e. Charlie, could see the vast array of what was happening from a molecular breeding approach and say, hey, here are a couple that might really work, instead of us kind of being vaguely aware of what everybody was doing. So,
Rick, you can't get tenure and promotion doing that kind of stuff. Everybody will smile and know that that's true. What you need is more of a person with a more of a bureaucratic orientation as part of the center to put all this information together. My admonition would be unnecessary and expensive duplication, especially in days like this when funding is cut so badly. We've really got to get out of our silos. But as individual faculty members, we used to have to run our own programs in order to survive and get funding. So Charlie can help a lot in creating the center. It's wonderful. But there has to be someone there with that specific responsibility. And it's really more of a bureaucratic type person who knows a lot about citrus.
Okay, that's fair, Chuck. Thank you. And I want this to be constructed. I'm not just ragging. I really wish we could find a way to just be a little bit more holistic in how we go about this. So that, Lucas, for example, Dr. Stolinsky, he shared with CRDF this discovery that he had made, that when the nymphs of psyllids feed on a tree that has this particular Bt toxin in it, it killed 100% of the progeny, killed them all, and killed all that same Bt toxin was killing Diprepes larvae down in the roots. So, I really appreciated Lucas sharing that with me. And, that was on a Friday afternoon, and I took the very unusual step of contacting the senior vice president of IFAS about it on Sunday night. I noodled on it all weekend and said, man, this is potentially really big. And the university flew into action. I mean, I appreciated the attention they gave it. But I don't know, it's just frustrating to me that there doesn't seem to be an integrated plan that tries to solve this problem once and for all. So thanks for letting me get on my soapbox. And by the way, Maria, we don't want to use antibiotics, but that was the only thing that we saw that could save the Florida industry in the time that we had left. And, I wish it were not that way. And there's a 180-day requirement from the time you inject the tree to when you harvest the fruit. So that antibiotic is long gone. You can eat that piece of fruit and not worry about it one bit. So, we fell into that because we had to, not because we wanted to. And marginal increase, marginal benefits is not going to save the Florida citrus industry. We need something that can reset the industry. And antibiotics seem to be the only thing to us that offered the hope of doing that.
Thank you, Rick. I really love all your comments, and especially because you make a point, and is we need to retrieve all the information that are in papers or are in documents that have been, done by other programs, and most of the time we don't know them. So I think reviewing that part, I think is key. And that happened also in FONTAGRO. We run project across all the Latin American region. We have 100 operations, and all of them project, produce publication and documents. But we need to go to the data that is there. So thank you for pointing on that. I really appreciate it. So now I want to go to Romina very quickly because we are running on time, and then I will pass the final words to Martin and then to Charlie.
Yeah,
Thank
very
you.
quickly, I just wanted to stress what Carrie said. Diagnostics and building capacity for diagnostics in regions that the disease is not present yet is definitely fundamental. And, the best strategy is exclusion, prevention, rapid eradication. Those are the best, strategies that we have. So for the regions that the disease is not present, definitely we should put effort in building capacity there and making those connections that they can easily monitor and surveillance for the vector and the pathogen. Thank you.
Thank you, Romina. Karen, you have your hand raised. I'm sorry, I didn't see it. Please.
Yeah, I did want to emphasize that Charlie has really been an excellent head on all of this. I'm not just saying this because he's my current guru, but he talks to all of us about the citrus. He may not have been an original citrus person, but he does talk to all of us. He does coordinate a major effort, and there are a bunch of new things that you folks can hear about tomorrow. That's it.
Thank you. Thank you very much. Oh, Rosemary. Yeah.
I would like to just say one thing, and that is that California has done a very good job of slowing up the citrus greening spread. And so the integrated management strategies that they're using might be a model, at least to start with, for other countries. In Florida, it's too far gone, and we never had the tools of being able to remove trees, whereas California has that, and that could be a regulation that either exists or could be put in place for other countries. So that could be, there are people who could speak very well to that issue, and that would be, I think, a very interesting discussion for people in the group who want to create an overall plan for reducing spread, immediate detection and reducing spread.
Thank you very much. Martin and then Charlie.
Well, just thank you all. Thanks to all the panelists, and thanks to the people who are watching live on YouTube. And for those perhaps that in a month or a year from now can see this webinar and get inspired to do something and to help with agriculture. I hope not with this problem, because this problem will be gone, but with another problem that we would sure have in the future. So don't miss tomorrow. We'll have 2 new panels, one on genetic control and one on coordination, international coordination. We'll meet at the same time tomorrow. So Charlie?
Again, thank you very much, everybody. I will leave with Rick's call to action to have a master plan. I've been doing a little bit of my part best I could. I'm not giving it away despite tenure. I think the future of science is not in individual labs, but across disciplines. That's where I think we are going to find a solution. And stay tuned for tomorrow. I think at the end of this workshop, we'll come with a collective mind to start building on Rick's master plan. Thank you very much. I'll see you tomorrow.
Thank you. Thank you very much.